Okay, we're going to continue on now with our action potential story. We're going to talk about how an action potential is propagated, which means that it is going to spread from one location to next down our motor neuron. If I can get the slide to advance. Okay, so as we mentioned in the last video lecture, the action potential is a temporary flip-flop of the charge difference across the membrane from -70, where it is at rest, up to +30. And that happens temporarily, and then it happens along a little local area of the cell membrane, and then we've got to reverse it and go back to normal and reset. And that happens through sodium ions moving in and potassium ions moving out to repolarize our cell membrane. Then we also said that this is going to start here at the axon hillock, that little widened area of the axon where it connects to the cell body. All right, so we need this to spread down the axon. So what happens is, let's say over here in the axon hillock, okay, let's say you've had an action potential generated here. So in that case, the outside becomes temporarily more negative. The inside is temporarily more positive. All right, now let's say over here, oops, hang on. All right, let's say over in here, we want the action potential to spread to that location. Okay, so over there right now, the inside is at its normal state. You know, everywhere else along the axon, the inside is more negative and the outside is more positive. Okay, well, what happens is when you do an action potential over here at the axon hillock, you've got sodium ions that have come in. All right, and ions will travel in currents. Those sodium ions are attracted to these negative charges you have over here. Okay, so guess what's happening? What's happening over in this area? Okay, as these sodium ions flow over here into this next stretch of the membrane that we have, what's happening to your membrane voltage? Guess what? It's starting to go up. What's going to happen if it hits -55? You get enough of those sodium ions over there, you're not going to be at -70. You're eventually going to hit -55. That's your threshold voltage. So over here in this area, you've got those voltage-gated sodium channels, and when you hit -55, they're going to open up, and then we're going to have sodium ions out here that come rushing really quickly inside the cell. And so here in that little patch of the membrane, guess what? Boom, we're going to have our action potential. We're going to go up to +30, and then just like we saw in the last video lecture, we're going to do our resetting process. So the voltage-gated sodium channels are going to close. The voltage-gated potassium channels you have there in that local area are going to open up, and you're going to reset the membrane. All right, now meanwhile, you've had these sodium ions that came in at this location. So guess what? They're going to spread over to the next stretch of membrane. You're going to have voltage-gated sodium channels over here. As soon as you get enough of those sodium ions over there, boom, sodium channels are going to open up. Sodium ions are going to start rushing inside, and you're going to go through the whole action potential process over at that next stretch of cell membrane. So that's how an action potential spreads, and that's also called propagation. Spreads, or it propagates in a wave down the axon. Okay, so as it spreads to the next location, you know, you've repolarized over here, and it's going to spread to the next location, and this area is going to repolarize, and then it's going to spread to the next location, and this area is going to repolarize over here. So it travels in a wave in its one direction all the way up and down the axon. So kind of neat how all of that works. All right. Now, if what makes this happen really fast? Sounds like, well, this is kind of a complicated process, and I mean, even though you do this depolarization in a fraction of a second, you know, we said earlier that some of these axons can be several feet long. They might be extending all the way from the spinal cord all the way down to, you know, muscles around your ankles and your feet. So how in the world are we going to get that signal almost instantaneously that far away? This is where something called myelin comes in. Myelination of axons is very important for action potential conduction velocity, which refers to how fast the action potential is going to travel down the axon. Okay, so let's back up over here for a second. So if we remember when we were going over the parts of a motor neuron, motor neurons have these little oval cells that live all up and down the length of the axon. We said those were called Schwann cells. And what Schwann cells do is what they do is they produce excess cell membrane in a sense, and then that excess membrane is wrapped up almost like a blanket around the axon itself. It creates a sheathing that covers the axon. But notice though that you have little gaps. So if you look at diagrams of axons of motor neurons, we talked about that earlier, but you've got little gaps there. Each of those gaps is called a node of Ranvier. Ranvier was the French guy who discovered these. So in those little gaps, you don't have the covering. You have open cell membrane there along the axon. So what really happens when you start an action potential here in the axon hillock, the action potential doesn't really occur in this type of neuron at every single location up and down the axon. It only happens where you have those gaps. So you're going to have your action potential here, and then it actually jumps to the next gap where you have the uncovered axon. Then it jumps to the next, then it jumps to the next, and then it jumps to the next, and it jumps to the next. And it's not really jumping like outside the axon and back inside. It's flowing inside the axon, but the depolarization wave goes super, 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 super fast from one node to another there inside the axon. The covering that you have here, made by those Schwann cells, is really functioning like the insulation you have around electrical wires. Insulation around electrical wires prevents leakage of the charge that you have in an electrical wire. And the same thing is happening with these sheaths that the Schwann cells made for us. Okay, so these sheathing, this insulation that those Schwann cells made for us, it's called myelin. It's a substance called myelin, and it's made from lipids and proteins like cell membranes are. Okay, and this, and again, it functions like insulation, and the conduction, the movement of the action potential down the axon, where it jumps from node of Ranvier to the next node of Ranvier to the next, is called saltatory conduction. That's another term for that. So the only place where you have those voltage-gated sodium channels that you need for your action potential will be at the nodes of Ranvier. So it really looks like the action potential is just jumping rapidly from node to node to node. In reality, it's just that the current, the change in charge with those sodium ions just rapidly progresses in between those nodes. And again, you'll see more about that. Your textbook authors have a very nice animation to show you how that works. This diagram from your textbook is giving you another illustration of how this is helpful and how it affects the speed of an action potential. All right, so if you had in parts of the motor neuron where you just have a typical plasma membrane, and you don't have any of those voltage-gated sodium channels, you're not going to generate an action potential there. You might have a depolarization there, but you're never going to achieve an action potential. And you've got all those leak channels, the sodium and potassium leak channels that you learned about on the resting membrane potential video, and those are just going to wind up resetting your charge difference across the membrane. Okay, now if you have the myelinated axon, like we were just talking about, you've got those. This is a representation of your a myelinated portion of the axon there. So the blue stuff is the myelin insulation with the axon on the inside, and then here's another myelinated part of the axon. And then right there you've got one of those nodes. So within the node of Ranvier, or Ranvier, those little purple things there are your voltage-gated ion channels that let you do your action potentials and reset them. Okay, so action potential will move very quickly along a myelinated axon as it jumps from node to node. Now, not all neurons in your nervous system have myelinated axons. Some of them are unmyelinated, and so they have voltage-gated channels all up and down the entire axon. So the action potential literally has to occur here, and then 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 here, all the way from one end of the axon to the other. So guess how that's going to affect the conduction velocity. Your action potential is not going to spread nearly as quickly down an unmyelinated axon as it will down a myelinated axon. And for some types of neurons, we'll learn more about this when we get to Unit 5, that's okay, they're supposed to be unmyelinated. But in the case of the motor neurons that control your skeletal muscles, you've got to have very fast communication. It's very important that our skeletal muscles respond really quickly from the signaling that occurs. And so you've got to have the myelin there in order for all of those things to work properly in a coordinated fashion. If you don't have, there is a disease that affects myelination of your neurons, and it's called multiple sclerosis. I'm sure most of us have heard of that before, or MS. What happens in MS patients, their immune systems are actually attacking the myelin coatings around the axons of their neurons, and that slows down the signaling that takes place, and that can interfere with their muscle coordination and other types of things that require nervous system communication as well. All right, so we've heard our really long story here about how a motor neuron, what it goes through to generate an action potential nerve impulse, but not losing sight of the big picture here. We're doing that so this motor neuron can signal a muscle fiber to contract. So we're going to, how in the world does that signal lead to, within the skeletal muscle, those thin and thick filaments, the actin and the myosin filaments, making those cross bridges that they need to make in order for the muscle to wind up contracting. So we're going to continue on with our story. We're going to hear what goes on at the neuromuscular junction in the next video lecture.